Hello, my name is Aidan Chalnick, and my project explores the relationship between the evolutionary history of Pseudomonas aeruginosa and its future potential to evolve antibiotic resistance. The current bacterial treatment paradigm is built around mutation rate and a drug's effectiveness in killing off multi-drug resistant organisms. However, the genetic processes that confer resistance are far more extensive than just mutation rate. The potential for multi-drug resistance is also influenced by a strain's current genetic profile, which is oftentimes a consequence of its source. For instance, a strain of P. aeruginosa inhabiting the ocean and a strain inhabiting a cenote pool face different environmental pressures which select for the regulation of differing alleles. In either case, a strain's source will affect its genomics and individual phenotypes. Consequently, an altered genome will place bacteria of the same species on different parts of the fitness landscape, restricting evolutionary potential. Failure to take this into account is a large part of why the current bacterial treatment paradigm is lacking. There currently exists no model for how specific environments affect resistance evolution. Although some data exist regarding common sources such as the cystic fibrosis lung, the influence of other environmental pressures on genomic alteration is largely unknown. In this experiment, antibiotic tolerance data was used in conjunction with the strain's previously sequenced genome as well as its source to determine how a strain's evolutionary history affects its future potential to evolve resistance, and what genomic indicators could have been used to predict specific resistances prior to any treatment altogether. This research serves in the preliminary identification of environmental effects on resistance evolution. Analysis of the effects of antibiotic passaging corroborates other findings, but also sheds light on the larger concept of environmental effects on observed genetic phenomenon. Future research into the evolutionary history of infectious bacteria is vital to reshaping the treatment paradigm. A logical next step would be research into how pertinent environments, such as the human lung, present conditions that provoke genomic adaptation. Studying factors such as interactions with other bacterial species is essential to understanding how environmental factors interplay with resistance evolution. This research will hopefully serve as an indicator of the importance of this work and that this current gap in knowledge must be explored further. Thank you.